Hi, this is Professor Amer Jamil. I'm working as Professor of Biochemistry in the Department of Biochemistry, University of Agriculture Faisalabad. My area is molecular biology, gene expression, DNA barcoding. Uh, this is a series of lectures. These are regarding the basic molecular biology. This is DNA replication first part, which contains some of the basics of DNA replication. Uh, what is DNA replication is, you might know that synthesis of DNA using DNA as a template, and this is a very important process which takes place in all types of living cells, which have to before cell division, because DNA has to be copied with fidelity so that new generation get the exact copy of the DNA. DNA replication is quite a complicated process, well tightly regulated, and this is quite obvious because if there is some mistake in the DNA replication, that will be translated in the form of mutation, and that can be lethal for the cell, leading to the cell death or some other complications. So here we will discuss about DNA replication in prokaryotes first, especially E. coli cells. DNA replication in E. coli, uh, this is the genome, E. coli genome, and these are the genes which code the proteins which are required for the DNA replication. So as you can see, a number of different proteins which are required for this process to occur, because as I mentioned that of high fidelity that is required. And there is, in case of prokaryotes, there is a single origin of replication, which is a sequence present on the DNA. I will talk about more later. But this is a single origin of replication that is present in case of prokaryotes, and a number of different proteins are there. This DNA replication is semi-conservative, and this was done in earlier experiment by Madison and Stahl. They used this heavy nitrogen, which is N15, as opposed to the normal nitrogen, which is N14. Uh, what they did was they grow the E. coli cells in the presence of N15 medium, so all the DNA that should contain N15, and then they perform cesium chloride density gradient. When they perform this, you can see the DNA is settled down at this place. So remember, it's all the nitrogens are N15, but when they grow the DNA, these E. coli cells in the presence of N14 medium, right very next generation, uh, those daughter molecules, they contain N14 and N15, a mixture of it. So, so then they uh, use this N14 for the further generation, and here similar results you can see. So there were three possibilities basically that this DNA replication is a conservative. If it was a conservative mode of replication, then we would see both of these strands of the parent strands and these new strands as a daughter strand. And in another case, it might have been like dispersive. In that case, we would see in this strand some part from the parent, some from the daughter. In both strands, it would have been like that. But this was not the situation. Here, what is the situation? Is quite straightforward. That you get one strand, which is a parent strand. As you can see here, this one is parent strand, and the other one is the daughter strand. This red strand is the daughter strand. So here the parent and the daughter. So this shows that DNA replication is semi-conservative. And similarly, in the next generation, when you grow again with the in N14 medium, so you can see from this particular parent, from this particular parent, you can see this one, like it contains one of the parent and one of the daughter. So these are again here one of the parent and one of the daughter. This time the parent from in this case is the red one, that was daughter in this case, is now parent in this case, and similarly with these two. So hence it was deduced that DNA replication is basically semi-conservative. And replication in case of prokaryotes, especially in case of E. coli, it takes place in theta mode of replication. As you can see here, this is a theta structure, and both of these strands, they start replicating simultaneously, and eventually you get this two different from one you can get two molecules, and each of these molecules of the DNA contain the semi-conservative mode of replication. They contain, one is the parent, another is the daughter strand. And another important thing is that DNA replication goes in the bidirectional form. So in this case, this lower one, this one is the bidirectional, and the DNA replication from where it is initiated, it is origin of replication, and the machinery which performs this function, that is called as replication fork. So there are two replication fork going on simultaneously in these two directions, whereas in this case only one unidirectional. So as you can see here, this these are the two spots which are given over here. This DNA replication starts in the bidirectional way, having two DNA replication forks. And the important thing is that DNA replication, there is a leading strand and a lagging strand. Why is that? Because this DNA replication is brought about with the help of enzymes which are called as DNA polymerases. And these DNA polymerases, they go in the 5' to 3' direction. So as you can see here, the new strand is synthesized in the 5' to 3' direction here, and in the 5' to 3' direction in this case as well. So when this DNA polymerase, it moves on the template, so DNA is a wound structure. As you can see, this is a wound structure. When this DNA polymerase, there is a movement of the DNA polymerase, this leading strand goes in the straight forward direction, whereas in the lagging strand, every time this machinery, when this DNA is opened up, DNA polymerization has to be initiated from the beginning, you can say over here. So here you get these types of fragments which are called as Okazaki fragments. These DNA polymerase on the, and this strand is called as lagging strand, where this DNA polymerization goes in the opposite direction, but its direction of synthesis is same, like 5' to 3'. So because both of these strands run anti-parallel to each other, this leading strand goes in the 5' to 3' in the straightforward way, and this goes in this direction. So you get these fragments, and named after the name of Okazaki, name of the scientist who discovered these. And this is the picture of Kornberg, who made some early discoveries regarding the DNA polymerases. And this is actually the mechanism of what is going on by the DNA polymerase, how these deoxynucleotides, they are added to the growing strand. So this one is a growing strand, and this is the template strand. Remember one thing, that DNA polymerases cannot initiate the synthesis of the new strand. They can only extend the already made strand. So in this case, you need some small oligonucleotide which is called as a primer. So in this case, first primer is synthesized, and that primer is synthesized by some other enzyme that I will talk about a little later. But in this case, you can see that there is a 3' hydroxyl of the DNA polymerase of the DNA strand. This is the 3' hydroxyl at this place, and this 3' hydroxyl has this lone pair of electrons, and this is the incoming triphosphate deoxynucleotide. And in this case, this this lone pair of electron attacks to this phosphorus. When this attacks to this phosphorus here, the electrons are shifted at this place, and this bond is broken, so pyrophosphate is released, and this phosphate makes linkage with this hydroxyl to make this kind of linkage, which is called as phosphodiester linkage. And this should be the complementary, the newly added strand, like this is T and this is A, so these both should be complementary to each other. And this pyrophosphate which is released, that provides energy for the DNA to move on the template. Here it is more clear, the mechanism of DNA polymerases. You can see there are 3 aspartates which are present here, 1, 2, and 3. And magnesium is required as a cofactor. Here is 2 magnesium, 1 is there, 1 is here. So you can see there is a linkage of magnesium, weak links, linkage of the magnesium with the 3 aspartates in the DNA polymerase. And the incoming nucleotide, deoxynucleotide with triphosphates, in this case same mechanism of action, and this one is the already present strand. So this is the oxygen, and this oxygen attacks at this, as I explained in the last slide, and this pyrophosphate that is released, and we see this is basically the phosphodiester linkage which is created here. You can see from the 3' hydroxyl, and this is the 5' phosphate of the next one. So in this way this strand grows, and continuously it goes on. One important function of the DNA polymerases is the proofreading, that if a wrong nucleotide is inserted, like in this case you can see this is a adenine, and there should have been thymine. In this case is the cytosine. So if this is a wrong nucleotide that is entered here, and this DNA polymerase, before it moves further, it removes this wrong nucleotide. Otherwise, as I mentioned earlier, there may be some mutation or something. So in this case, there is a 3' to 5' exonuclease activity that takes place. So this 3' to 5' exonuclease activity, that basically functions in this case for this proofreading activity. It's responsible for the proofread activity of the DNA polymerases. So what it does is it removes these wrong, mispaired, you can say mispaired deoxynucleotide. It removes that with the help of this 3' to 5' exoactivity, and then it adds the new nucleotide which is the correct one. So in this, with the help of its polymerization activity. So proofreading activity is very important for the DNA polymerases which is present in prokaryotes and eukaryotic cells. This is a relation of the DNA polymerase with the with its template, the DNA itself. This will come back later actually. This is very important, that what are the important functions of DNA polymerases. In case of E. coli, there are at least 5 different types of DNA polymerases, but the most important ones which take part in the reaction of DNA replication are the DNA polymerase 1 and DNA polymerase 3. This 2 is also important for DNA repair and other functions. So we will discuss here the properties of DNA polymerase 1 and 3. Here the structural gene is given is called as Pol A, and as you know that the name of the gene is written in lowercase italic form followed by uppercase italic, and this is Pol C for the DNA polymerase 3. In the brackets, DNA E is the older name of this DNA polymerase. In case of DNA polymerase 1, only one subunit of about 103,000 molecular mass, whereas in case of DNA polymerase 3, there are more than 10 subunits. It's a big protein, and this molecular mass, total molecular mass, is 791,500, which is very large as compared to DNA polymerase 1. And this Pol C is this is just for only one subunit out of those 10. This Pol C subunit corresponds to the subunit which is actually responsible for DNA polymerization. And this proofread activity, 3' to 5' exoactivity, that is present in all 3 types of DNA polymerases. However, there is a special type of DNA polymerase exonuclease activity that is present only in DNA polymerase 1, and it is not present in other DNA polymerases. It is the 5' to 3' exonuclease activity, and this 5' to 3' exonuclease activity, it is present only in case of DNA polymerase 1 in E. coli. It has some special function which I will talk about later. This polymerization rate, how many nucleotides are added per second? As you can see in DNA polymerase 1, it is 16 to 20 nucleotides. In case of DNA polymerase 3, it is 250 to 1000 nucleotides per second. Is quite fast speed. And the processivity is also an important factor, that for how long DNA polymerase can move on the template before it falls off. As you can see, DNA polymerase 1, there are 3 to 200 nucleotides. Its processivity is here. In case of DNA polymerase 3, processivity is very large. As you can see, more than 500,000 nucleotides before it falls off the template. And it was a requirement because DNA polymerase 3 is the major enzyme which performs this DNA replication, whereas DNA polymerase 1 is required for a specific function. It's not required, it's not the major enzyme of the DNA replication in case of prokaryotes. And this is the function of DNA polymerase 1, that its substrate is nicked DNA. Nick means when the phosphodiester linkage is broken. So here you can see this phosphodiester linkage is broken. So this is a nick is produced over here. So it may be RNA or DNA. So DNA polymerase 1 recognizes this sort of nick which is present here, and it starts adding deoxynucleotides, like it will add deoxynucleotide to this place at this hydroxyl, and new incoming triphosphate, its monophosphate will be added, as I mentioned earlier, that how this DNA polymerases perform their function, and this pyrophosphate is released. And this nick is it moves forward. This nick moves forward, and similarly it goes on and on with reference to the processivity of the DNA polymerase. So as you can see, nick was started at this place, and now nick has reached at this place, and this is basically, this is called as nick translation. So nick remains there. DNA polymerase cannot seal this nick. It can only replace a part of the DNA or RNA present in this case, but this nick cannot be sealed by DNA polymerase 1. There is another enzyme which seals this function. So it is now quite clear that the function of the DNA polymerase 1, that's why its processivity is low. We don't need very high processivity of DNA polymerase 1 because its function is does not relate to the major function of the DNA replication, but only the replacement of a small part of the DNA. If we look at DNA polymerase 3, is quite a complicated enzyme. As you can see here, there are more number of, these are the subunits which are present. There are different names, alpha, epsilon, theta, tau. Similarly, they have delta, delta prime, psi, phi, beta, and they have different subunits of these are number of subunits are present, like 2 alpha, 2 epsilon, 2 theta, 2 tau, and similarly these are the 1 and beta. There are 4 beta subunits, and if we look at the molecular mass, there are different sizes, and these are the names of the genes which are present here, and function of the subunits. The major, as I mentioned earlier, is the polymerization activity that is present in the pol C, and then there is a proofread activity, and then there is a stabilization. There is a clamp loading. We will talk about later what is that, what it means. And basically, most of the this is the core polymerase that contains polymerization activity and proofreading activity and some sort of stabilization of the subunits. But all the rest actually enhance the processivity of the DNA polymerase, because this high processivity is also needed because of the large size of the chromosome that has to be replicated in a short period of time. So all these different proteins like clamp loader, clamp opener, and all that, they are required for increasing the processivity of the DNA polymerase 3. And this is the cartoon of DNA subunits of the DNA polymerase. As you can see, inside are these beta clamp that are present, and this is also this is a beta clamp. Basically, these clamps are present. This is one clamp, this is the another one, and these clamps stabilize the DNA polymerase on the template. And this is contains the core. This is the core, and these are the two cores which are present in one set of DNA, and these are the tau proteins, and these are the other proteins, and this is the beta clamp which is present at this place, and then it is loaded on two sides. So this is overall structure of the DNA polymerase 3. And DNA polymerase, DNA B helicase, this is also needed. There are 6 subunits of DNA helicase because to unwind the DNA, which is a double helical wound structure. These are just different views of the DNA polymerase working on DNA template. Now, before going further, it's pertinent to discuss about the origin of replication. Origin of replication in case of DNA, in case of E. coli DNA, that contains different sequences present at a certain place on the chromosome from where DNA replication is initiated. So, as you can see, there are 3 13 base pair sequences which are present, and this is the consensus sequence, and these are present, and these are DNA unwinding elements. So these 3 which are present here in this place here, and then there are 9 base pair sequences having, if we look at the consensus sequence, this is the consensus sequence for these. This is the consensus sequence, and these are the 9 base pair sequences which are present a little apart from each other. They are not present in tandem. So these, and then there are some other like IHF, integration host factor, FIS, factor for inversion stimulation. So whole of this makes this DNA origin of DNA replication, where DNA replication may be initiated on the DNA. So these are the sequences, specific sequences which recognize the proteins of the proteins required for the DNA replication. And this is origin of DNA replication. This is model of initiation of DNA replication and E. coli region, and it is called as ORIC. ORIC is its name. And this is the DNA unwinding element, as I showed in the last slide. And this is the initiating protein. You can say this is a DNA A protein along with ATP. So this is a member of AAA plus ATPase protein family. ATP is associated with diverse cellular activities. So here its function is basically to unwind the DNA, which is at the DUE region. So this, with the help of this ATP, it unwinds. So DNA A dependent denaturation of the DUE region that takes place here. And then comes the DNA B protein and DNA C along with ATP. DNA C basically it helps the loading of DNA B. DNA B is a helicase which of course will be required, as you can see here, mentioned earlier in the picture of the DNA polymerase 3. These are the 6 subunits. So 6 subunits here, 6 subunits here. So there are 2 replication forks that will go on in opposite direction, and these are the 6 subunits each of the DNA B protein, which is a helicase. And this DNA C protein along with ATP helps loading of this on this region of loading of the DNA B protein, and it is released. DNA C and ATP is hydrolyzed in this case. And these are the proteins which are required for initiation of DNA replication at E. coli origin, and these are the DNA A proteins, as I mentioned, and these are the molecular masses and number of subunits of these proteins which are required for replication. Its function of DNA A, it recognizes the sequence and opens the duplex, and DNA B is a helicase which unwinds the DNA, and DNA C helps loading of the DNA B on the template, and this is HU, FIS, IHS, IHF. All these, they have different functions for which are required for initiation. And another important one is the primase, which is DNA G protein, and this DNA G protein basically functions for the synthesis of RNA primers. As I mentioned, that DNA polymerases cannot make primers, cannot initiate any change. So you need some other enzyme which is already there. So these primers are the small oligonucleotides which are first made with the help of DNA primase protein, which is also called as DNA G protein, and these primers allow the synthesis of DNA replication to take place, to continuously it goes on. And then we have SSB, single strand binding proteins, which are required for, so that the it binds the opened up of the two strands. If the strands are opened up, they should not be joined back. So this is the function of these proteins. And then DNA gyrase is a topoisomerase 2 type of enzyme, and this protein relieves torsional strain. And this DM methylase, it methylates the GATC region sequences at the REC. This function will come later as well. So as you have seen that these are some of the proteins, there may be some more in fact, but these are some basic proteins which are required for initiation of DNA replication. So making whole thing in one place. So here there is a leading strand and then there is a lagging strand, and this leading strand also initiated with a DNA G protein which is primase. So in this case RNA primer is synthesized from, and you will see a lot of RNA primers, more number of RNA primers on the lagging strand, because I mentioned earlier that these are synthesized in Okazaki fragments. So here you can see one of the primase activity is going on here, and this is RNA primer is synthesized here, and these are the single strand binding proteins which allow two strands separate, and DNA B helicase is there. So DNA polymerase 3 performs the major function on both the strands. So DNA polymerization goes in 5' to 3' direction in both the strands, and the torsional strain that is removed with the help of DNA gyrase or DNA gyrase, DNA topoisomerase 2 enzyme. This is a more clear picture of the same process I just mentioned, and okay, this one is the overall picture. As you can see here, it looked in the previous slides that there are two DNA polymerases which are moving in the opposite direction, and the one on the leading strand and another one was the lagging strand. Actually, this is not the situation. Actual situation is that there are two core of DNA polymerase 3, both are working simultaneously on the two strands of the DNA, both one on the leading strand, another on the lagging strand. So as DNA polymerase in both cases in 5' to 3' direction, then how is it possible that only one DNA polymerase moving in one direction can perform the function of polymerization on the leading and lagging strand simultaneously? In fact, this happens because there is a loop in the DNA polymerase, in the DNA itself. So you can see this is a looping that takes place. So this looping basically, it allows the DNA, the lagging strand of the DNA to be placed in front of the this DNA polymerase. So when because of this loop, this DNA is turned around basically, and so that this whole of this set is moving in one direction. And this other process is same, that there is an addition of primer with the help of primase and other proteins, and one important function is of the clamp. So you can see in all these pics over here, this is a beta clamp here, and this is a beta clamp. In both cores of the DNA polymerase polymerase 3 contains this beta clamp, and this is beta clamp loader. As I mentioned in the table, that there are different functions, especially this beta clamp loader. Why is it needed to move along with the DNA polymerase? Because in lagging strand, so there are Okazaki fragments, so every time it moves further, the older clamp is discarded, it is removed, and new clamp has to be loaded on this place. So that new clamp comes from this, and this is the clamp loader. It loads the new clamp at this place, and the old clamp is discarded. So this process is continuously it is going on. So here is a more clear picture. This you can see, this is the uh, clamp loading complex, and these are the clamps, and this is the loop you can see. So both of these moves in the same direction, and 5 prime to 3 prime direction, but in this case, because of the loop, this DNA polymerase looks to be moving in the same direction. And this clamp, when it is used, this primer is primase is also released, and it is also a continuous process, especially on the lagging strand, because of the Okazaki fragments. And there is a discarding of the beta clamp and addition of the new one here. So this continuous process, it takes place, and this process continuously going on. So basically these are the proteins which are required at the E. coli replisome, and this is the functions of most of these proteins. I already have mentioned the single strand binding protein, DNA B protein, which is helicase, DNA G, which is primase, polymerase 3, and then this DNA polymerase 1 removes the RNA primer, and DNAligase to remove the torsional strain, and DNAligase is function is to seal the nick, which is finally it is present over there. So here is the nick, which is translated because of the removal of this RNA primer with the help of DNA polymerase 1, and DNAligase is the enzyme which seals this final nick by using ATP or NAD plus as cofactors, and this DNAligase seals the nick. So this process is completed. So this is the mechanism of action of DNAligase, and this enzyme, it contains a basic amino acid lysine at its active site, and this NH2 group that is first, this enzyme is activated with the help of addition of AMP, adenosine monophosphate. It may come from ATP or it may come from NAD plus. So if it is coming from ATP, pyrophosphate is released, and then AMP, it comes here. So this is AMP. So this is called an enzyme AMP complex. And if it is coming from NAD plus, so NMN is released. So in both cases, AMP is added at this place, and this is the activation. And this is the nick, as I mentioned, that it is only a one bonding is missing in the phosphodiester linkage. So this activated enzyme, it helps in the attack of this electrons to this phosphate, and this is sealed, nick is sealed, and this AMP, which actually came for activation of the DNA ligase, it is released. So what is going on overall is that DNA ligase is first activated by the addition of AMP, which helps in the formation of this phosphodiester linkage, which is present at the nick, and AMP is released. So this is a completion of DNA polymerase activity over there. And the how this termination takes place, these are TER sequences which are function. These are terminus sequences, and TUS, there is a protein which is called as terminus utilization substance, and this is the region of replication. Of course, this is out of scale, and two forks, one is going on in the clockwise, another is going on in the counterclockwise direction. And when they hit to these sequences, which are termination sequences, there are different A, B, C, D, and this is A, B, D, C, D, G, and there are two forks when come, one from the counterclockwise, another from the one clockwise direction, they came in this place. As you can imagine, these are the speed breakers, so they slow down the DNA polymerases over there, and one of those is trapped inside these sequences, and other one finally reaches to the sequences, which is halted one. And then what happens is that both of these, this is the place where both of these sequences, they are present at this place, at this junction. These are new strands, these are two new strands, and counterclockwise and clockwise they are intertwined with each other. So this is called as catenated chromosome. DNA topoisomerase 4 enzyme is required to release these two chromosomes separately, which can go in the daughter cells. So with this I end with the replication, major part of the replication in case of prokaryotes. And for the eukaryotic DNA replication, there are some differences that I will discuss in my next part of the talk. I am thankful to Leininger's Principles of Biochemistry, from where I got this whole of these slides from there, so that the students may understand it well. Thank you very much.